genome evolution is a topic, then we need to start off with the definition of the genome. And by the end of today, I'm hoping that everybody will be comfortable using experimental techniques, or at least proposing experiments, to test hypotheses about how genomes evolve. So specifically, something that will be familiar to those of you that took genetics with me, chromosome fissions and fusions, and how those are involved in chromosome evolution. So here's a picture. I always start off with defining what we're talking about. Here's a picture that defines the human genome. So if you described in text or verbally what the genome is, how would you define a genome? What is the human genome? All of our genetic material? Okay. So what's missing here from the human genome? We've got 2 copies of every chromosome, 1 through 23, the X chromosome being the 23rd. So it's, maybe missing the Y chromosome. So that would be representative of the male genome, 1 X and 1 Y chromosome instead of 2 X's. What else would be missing? All of the DNA in a cell. Yes, the mitochondrial genome would be missing as well from this picture. So if this really is the human genome, I would add that you want to have the circular mitochondrial genome as well. And depending on who you're talking to, they might or might not insist on having a Y chromosome there as well. So what features about our genome can you see in this picture? If you were going to describe our genome, what would you say about it?
It's not all the same.
So what parts of the genome aren't the same?
So as far as the chromosome is concerned, like chromosome 1 would be like they have like homologous pairs. They look a little bit different.
So should they look similar or should they look different? We've got 2 copies of chromosome 1. They should look similar. They're homologous, as you said. So we have homologous pairs of chromosomes, homologs. So we've got pairs of chromosomes. The human genome is diploid, 2 copies of each chromosome.
When I'm looking at, say, chromosome 1 compared to chromosome 20, there's different sizes.
Okay, so there are different sizes of chromosomes. Chromosome 1 versus chromosome 20, for example. So we've got a variety of different chromosome sizes. What else could we use to describe the human genome? All of these things can evolve, of course. So sizes of chromosomes can evolve, the number of chromosomes can evolve, change, that is, between populations, between species. These are the sorts of things we'll be talking about when we talk about genome evolution. What feature of a chromosome can't you see here that sort of defines types of chromosomes or classes of chromosomes? Yeah, so where the centromere is located. It's very difficult to see on these particular metaphase chromosomes where the centromeres are. But that'll become more apparent in some future slides. So we've got a lot of different aspects of the genome that can evolve. All of this is DNA, of course, and all evolution is changes in DNA over time. So if we have a change to our chromosomes, we have genome evolution. Drawing from one of my favorite resources, Wikipedia, which is always legitimate. If you cite it, cite the primary reference, not Wikipedia itself. But so right here, for example, there are some primary citations. If you wanted to go talk about chromosome evolution, for example, you've got links to the actual primary literature. So this is really striking, and this is what the textbook talked about in case you happen to read the chapter already, for the first 2 sections. This paradox between complexity and the number of chromosomes or the size of the genome. So at the very low end of the number of chromosomes that any species could have, talking eukaryotic species in this case. We've got the jack jumper ant, a whole whopping 1 chromosome, or diploid, 2. So 2 chromosomes means 1 pair of homologous chromosomes. So it has 1 chromosome. Chromosome 1, that's it. Then you've got some mites that have between 4 and 14 chromosomes. Mosquitoes have 6, etc. What do you think is the upper end of the range of number of chromosomes organisms can have? Obviously, you have to have at least 1. That's the jack jumper ant.
I know I've heard like goldfish or something has like 72 chromosomes or something.
So goldfish have a large number. You said 72. I think actually on the next slide we find out something about that. Any other thoughts? Does anybody know about patterns of chromosome number in different groups of organisms?
Those guys have 20?
Yeah, so-
4 chromosomes in one, or something like that?
Right, a lot of some organisms are tetraploid. You've got 4 copies of every chromosome. So this is diploid chromosome numbers. You could talk about plants. There are some fish that are tetraploid. There are some plants that are octoploid. They've got 8 copies of every chromosome. Some strawberries are dodecaploid. They've got 12 copies of every chromosome. So there's a lot of plasticity, variation in genome size, how much DNA you have, and also how many chromosomes that DNA is divided up among. So goldfish is kind of related to a carp. They're kind of close relatives. 104 chromosomes, so that's what, 52 pairs? Okay. Then you've got the adder's tongue, which, if I remember correctly, is a fern. Don't recall offhand. 1,440 chromosomes. So what, 720 pairs? I pity the geneticists that had to actually go in and count accurately. And this is why there's not a lot of information known about the upper limit, because sometimes you look at the chromosomes and say, holy shit, that's too many to count. Move on. Because it's really hard to distinguish that many chromosomes that are in a single nucleus. So why aren't there lots of organisms with all this number of chromosomes? Right? This seems, on this list, which is certainly not a complete list, this 1,440 sort of seems like an outlier. 94, 104, right? Not every species is listed. And then this big jump. So is there an upper limit? Is there a theoretical upper limit to how many chromosomes you could have? What sort of forces or what sort of resources, what sort of evolutionary pressures would come into play when an organism is selecting for or against having yet 1 more pair of chromosomes?
Well, you have to like replicate all that every time, right? I mean, so that's a lot of energy that you're going to be spending the more you have to replicate. So I guess depending on the organism, if you have a lot of energy to do so, then maybe there's not much of an upper limit, or the upper limit's a little bit higher than some other organism.
Okay, so one idea is how much DNA do you have to replicate? Now it's not, what we're not seeing here, the information that would be really useful and has to do with the C-value paradox, which is talked about, or G-value, depends on who you're talking to, is a relationship between the number of chromosomes and the size of the genome. So we're just talking about chromosome number here. So it could be that the jack jumper ant has 1 big chromosome, and that Ophioglossum has loads and loads of tiny little chromosomes. So you'd have the same amount of DNA, and I'm not saying you're wrong, I'm just saying this is another point to make. That yes, the number of chromosomes, there might be an upper limit, but also the size of the genome as a whole certainly has to do with, how much DNA do you have to replicate. So there's an upper limit guaranteed to how big your genome could be. There's a point at which you have a cell that doesn't have the resources to actually make a copy of all of that DNA, or maybe even pack all of that DNA into the nucleus.
You have just touched on mine, nuclear space.
Oh, fabulous. I'm sorry to intrude, but yeah, absolutely. And so is there an upper limit to the size of the nucleus? Is there an upper limit to the size of a cell? How big can cells get? Well, tumors are multiple cells, but yeah, you can, well, ostrich, an egg is a cell. Think of how big an ostrich egg is if you've seen one. Yeah, they're like basketball sized or maybe a little bit smaller. That's a special type of cell though, but there is an upper bound to the size of a cell, and there's an upper bound to the size of a nucleus. But what about chromosome number? So in terms of resources, what sorts of forces limit the number of chromosomes you have, if not the amount of DNA?
Isn't it the way that the cell divides? Because I know that plants have a lot, but it's because of the way that they replicate the cell.
So there might be a difference between kingdoms, for example. Plants might have a benefit being able to replicate, and that's why we do see a lot of plants that are tetraploid, octoploid. They've evolved ways to segregate all of those chromosomes accurately. So yeah, if you're right, humans, if you've got one extra copy of a chromosome, that's a whole separate discussion, trisomy, and how that affects individuals as well. So some of the points I'm thinking of about what dictates the number of chromosomes might be, how do chromosomes physically segregate at meiosis and mitosis? What sort of resources does a cell need to actually pull the chromosomes apart? So you need mitotic spindles, you need microtubules that grow from the asters, the ends of the cell essentially, and then grab all of those centromeres. There might be a limit to the number of microtubules a cell can build to attach to all of those, think about 1,440 centromeres.
Some of them don't get attached, right? Some of them just go from one to the next.
Presumably, I don't know the answer to the question of how accurately this happens in organisms that have large numbers of chromosomes, but presumably it happens accurately because of the bad things that happen when you get aneuploidy. So in humans, if you have non-disjunction of a chromosome, if they don't segregate exactly 50-50, that causes things like trisomies, Down syndrome, and so forth. And the same thing is true for other species as well. So when you see an organism in nature that has that many chromosomes and it seems to be functioning fairly well, that suggests, to me at least, without scientific evidence, that chromosomes are segregating faithfully, or at least the organism is able to tolerate inaccurate segregation. This part of the table makes another point about the C-value or G-value paradox, the relationship between the number of chromosomes or the genome size, and in this case, complexity. And I hesitate to say complexity as an evolutionary biologist because it's difficult to say whether or not a radish or a kangaroo is more complex. I think a radish has a more complex flavor, but radishes have more chromosomes than kangaroos. And the more interesting point is, I just picked this as a random example, but then I went to look at how many chromosomes all kangaroos have. Kangaroo is kind of a big category. There are a number of kangaroo species. But not every kangaroo species has the same number of chromosomes. So even between closely related species, you can have vast changes, for example, from 16 to 40 chromosomes within a single genus. So closely related species of kangaroo can drastically differ in the number of chromosomes they have. Evolution, change in genetic content, in this case, the number of chromosomes. You think number of chromosomes is something that can be selected for or against? Like, is it better to be a kangaroo with 16 chromosomes or 40 chromosomes? Why would it matter?
Maybe it depends on the genes that are on those chromosomes.
Certainly could depend on, what genes are on the chromosomes, which chromosomes were involved. So one of the things we're going to talk about today is how you go from a small number of chromosomes to a larger number of chromosomes. So how do you evolve more chromosomes, or in the opposite, how do you evolve fewer chromosomes? And both of those things happen in nature. So some species actually reduce the number of chromosomes by evolution. Other species, there is an increase in the number of chromosomes. It depends on this particular case you happen to look at. So at the very basic level, the concept is that chromosome numbers can change, as can the genome size, but today I'm focusing more on the number of chromosomes. And one of the ways that chromosome number can change radically is this process called whole genome duplication. And it's just like it sounds. I'm not going to dwell on this point. It's simply a cell happens to replicate all of its DNA twice instead of once. So you have a diploid cell. It copies the diploid number of chromosomes. That's the genome duplication part. So you're temporarily tetraploid. This organism that was a diploid organism becomes tetraploid. This is how a lot of plants, and especially salmon also became, and a lot of fish became tetraploid. Your whole genome copies itself one extra time. So then you've got 4 copies of every chromosome. Beneficial or detrimental, do you think? Don't read the bottom text, that'll give it away. So it can be beneficial. Could be detrimental, so resources, you've got twice as much DNA now for a cell to copy if you're tetraploid. So that could clearly be detrimental. It takes a lot of energy to copy that much DNA. You have to have all the nucleotides, you need to synthesize a whole extra copy of the genome in every cell. But the reason that evolutionary biologists think that this is something that occurs, that we observe so many species that are tetraploid, octoploid, 8 copies of every chromosome, and decaploid, dodecaploid, is that having that many copies of every gene in the genome means natural selection has something to play with. Like natural selection's playground. You've got 8 copies of every gene, and you only really needed 2 copies of every gene to be healthy. Then you've got, presumably, this is generally true, 6 extra copies of genes that can have mutations occur in them. And those mutations might slightly tinker with how well that gene, protein functions in the environment that the organism finds itself in.
Question, so
Please.
when they replicate the second time, they still remain tetraploid, right? They don't become quadruploid?
Right. So the idea is that there's one genome duplication. From that point forward, the cells are fine with having 4 copies of every chromosome to replicate. And that genome duplication doesn't happen again, necessarily. And there are really complicated models that evolutionary biologists have devised to explain from cell to cell how this happens. So when cell division happens, what happens to this extra set of chromosomes? And how do you wind up, for example, the biggest question that a lot of evolutionary biologists need to deal with is, if a whole genome duplication occurs, are these individuals able to hybridize with non-genome duplicated individuals?
Isn't that what happens in the seedless watermelon?
You've got,
Go from tetraploid to diploid.
well, it's beneficial, and I'm not sure about the case of watermelons. Do watermelons self, or are they obligate outcrossers? Something that I don't know about, but I can definitely look up. Quick, Wikipedia, help me out. So this is a benefit of, this might be why you see so many plants, if they're selfing plants, that can sustain whole genome duplications. The issue being that if you've duplicated your whole genome, if I happen to be tetraploid, which I don't know if I am, I suppose I'm probably not, but if I were tetraploid, then presumably I'd have to find a partner to have kids with who is also tetraploid. Because if I was tetraploid and I mated with a diploid organism, our kids would be we would be probably triploid. So if you've got a tetraploid organism, 4 copies of every chromosome, after DNA synthesis, this is the start of meiosis, then you've got 8 copies of the chromosomes, and you go through the first and the second divisions of meiosis, The first division, you get 4 copies of every chromosome in the 2 daughter cells, and then each of those divide. In the second division of meiosis, you wind up making gametes that are diploid, which is what you'd expect of an individual that was tetraploid. Meiosis will still work the same way. You still get 2 divisions. So you go from 4 chromosomes, you replicate through synthesis to 8, and then you divide and divide. 8 divided by 2 divided by 2. And if that individual is going to mate with a normal member of the species, so if this individual had a whole genome duplication and it was trying to mate with an individual that hadn't gone through a whole genome duplication, then after meiosis, you are forming a triploid individual. So you've got a gamete that's got 1 chromosome joining together with a gamete that's got 2 chromosomes. And then you've got this really weird situation, which what do you do if you've got 3 chromosomes? If you have, so you could play with, if you want, if you have the motivation, how does meiosis in a triploid individual work? So do you synthesis, division, division, and then how many chromosomes do you wind up in every haploid gamete? Or not necessarily haploid. How many chromosomes do you wind up with?
One and one, others.
Right, so if you're triploid, 3 chromosomes, after synthesis you've got 6, and then you undergo 2 rounds of cell division. So it's an average of 1.5 chromosomes per 4 haploid, well, I don't know what the word is for haploid and a half. There is no word for haploid and a half because it doesn't happen. So yeah, maybe half of the gametes get 1 chromosome and the other half of the gametes, the other 2 products get 2 chromosomes. I don't know. So anyway, boy am I long-winded this morning. This is pretty fantastic too because I woke up at 4 and I was already tired by the time class started, but now I'm getting invigorated. So triploidy, not very good, bottom line.
Is it possible with the whole genome duplication that natural selection can actually select against it and it revert back to its original state of diploidy or whatnot?
Right. With the concept, the question is it possible that natural selection acts against whole genome duplication? Absolutely. Especially if you consider the idea, which I probably should have explicitly stated, that whole genome duplication happens in one organism. And so if that organism isn't able to produce viable, fertile offspring of its own when mating with whoever it mates with, then that organism that had the whole genome duplication doesn't pass on the whole genome duplication to the next generation, so its fitness is 0 and it goes extinct. But if you're a selfer, to go back to the plants just for a second, then you mate with yourself. So if your pollen and your eggs are both whole genome duplicated, then you can have offspring. And that might explain part of why a large number of selfing species tend to be are able to sustain whole genome duplication. How it happened in fish? So one of the great examples that's been well studied of chromosome number change, the evolution of chromosome number, occurs in us. And of course, this is sometimes controversial, so I enjoyed talking about it in class. What are the relationships between non-human primates and us, the human primates? We've got a different number, so we're special. We've got a different number of chromosomes than the non-human primates. We've got 46 chromosomes. Our closest relatives, chimpanzees, orangutans, and gorillas, have 48. So they've got 24 pairs, we've got 23 pairs. So we're special, we're different. But it's important to know how and what the evidence is that relates us to those non-human primate species. So here are a couple of 3 pairs of ideograms, drawings of the chromosomes of these different species. Bless you. So this is just a drawing of the chromosome banding patterns. You treat the chromosomes with chemicals that elicits these dark and gray bands to appear. And this is some of the earliest evidence, not the earliest evidence, that humans and non-human primates were close relatives. So cytogeneticists looked at the banding patterns and said, hey, those look pretty similar. And this is, in this example, just looking at chromosome 2. So our chromosome 2, shown on the left, looks a lot like what would happen if you added a chimpanzee chromosome that has pretty much the same banding pattern as the human chromosome to the P arm, the short arm. And then our Q arm of our chromosome 2 looks an awful lot like these other chromosomes that are also present as separate chromosomes in chimps, gorillas, and orangutans. So this suggested that in the lineage leading to us, 2 chromosomes that were present in the ancestor, not modern humans, but the ancestor of us and chimps, gorillas, and orangutans, in the human lineage, those 2 chromosomes stuck together, they fused. And these sorts of changes happen all the time, and they're not necessarily selected for or against a change in the number of chromosomes from, say, 24 pairs to 23 pairs. I mean, what's essentially different between chimpanzee, gorilla, and orangutan chromosomes compared to the human version of the fusion chromosome? What's different in terms of the genome? That is, focus on chimpanzee, for example. If the chromosomes in chimpanzees broke there and attached to each other, what changes? What's different between chimpanzees and humans? Those 2 chromosomes break right at their tips in chimpanzees, and the 2 long arms stick together, form a big fusion chromosome. Does the amount of DNA stay exactly the same from chimpanzees to humans? So what happens? What happens to this tiny little fragment of the P arm of the chimpanzee chromosome and this little fragment down there that aren't part of the fusion? They get lost. They disappear because they don't have a centromere. They might get replicated during the first cell division, but if there's no centromere, there's no way for them to segregate properly at mitosis or meiosis, so they get lost. So presumably any chromosome fusion, with rare exception, involves a little bit of loss of DNA. So decrease in genome size, and if there are genes important to the organism in those regions, then you might expect it would be selected against.
I'm sorry, what did you say about the genes in those regions?
So if there happen to be genes, if there was an important gene, like, I don't know, DNA polymerase, found on that tip of that chromosome, when that fusion chromosome gets produced, just the 2 little tips of those 2 chromosomes, something like that, if that chromosome carries an important gene, but it's not able to be faithfully separated, segregated into daughter cells during replication because this chromosome has no centromere. So it's got nothing for microtubules to attach to, so it's got no way to be pulled apart. One copy goes into that daughter cell, another copy goes into that daughter cell during mitosis. Then those genes start getting lost from particular cells in the lineage of that organism. And that would presumably be bad and get selected against. But the fact is, we do witness changes in chromosome number that occur by chromosome fusions. And when chromosome fusions happen, often there are these little tiny fragments of DNA that get jettisoned, essentially, as long as they're not carrying a gene that's important for the health of the organism. Questions or concerns about this concept? I know it's a strange one, but chromosome fusions and the opposite fissions, breaking of chromosomes into multiple chromosomes, happen pretty frequently. So here's not the ideogram version, but the actual karyogram version. So looking at the actual chromosomes, banded chromosomes, of the 4 different species, from left to right, shown down here, humans on the left, then chimpanzees, then gorillas, then orangutans. So we've all got the same number of chromosomes, except for those 3 non-human primate species have 2 chromosomes where we have 1. Right? Chromosome 2. And the banding patterns of those chromosomes look very similar to each other, and that's what suggested to cytogeneticists that humans are basically fancy primates, or primates are fancy humans, depending on which way you want to look at it. But of course, extraordinary claims like we're closely related to chimpanzees require extraordinary evidence to support it. So not only was the banding patterns of the chromosomes, which are unique, and so suggest the relationships between our chromosome 2 and primate chromosomes, we had to do something else to be really convincing. I'm not exactly sure why I put this in. I guess it's to show the centromere locations, because this is exactly the same, it's just a different version of the same karyogram. So you can look at chromosome 2 and see that, how would those 2 chromosomes fuse together? So assuming in the common ancestor of humans and chimps, it was those 2 chromosomes that fused to create what we have as one larger chromosome. So would they fuse at the centromeres? Possible. How much chromosome do you lose if these 2 chromosomes fuse at the centromeres? Yeah, it depends on which part stays and which part goes. So it could be, one possibility is that the short arms of both of those chromosomes don't retain centromeres and they get lost. But another possibility, which is also possible, if you have 2 submetacentric chromosomes like that, where the centromere is sort of off-center. If I draw the centromere as a red bubble, you can have chromosome translocations, not just fusions, where 2 chromosomes stick together and that's it. If those broke exactly in the middle, which I'm not drawing exactly in the middle, I'm trying, and exchanged parts, so there's a fusion there, you might wind up with the 2 long arms, the Q arms sticking together, and you might get a tiny little chromosome. But each of them would still have a centromere. If the chromosomes break at the centromere, like right in the middle of the centromere, every arm, the long arm and the short arm, is still attached to part of the centromere. And when they come back, when they fuse, if they fuse like this, then you wouldn't have a change in chromosome number. You just have a change in chromosome sizes. You start out with 2 chromosomes that are fairly similar in size, like here, and then you'd wind up with 1 larger chromosome and 1 smaller chromosome. So exactly the same genetic content, no loss of genes, no loss of DNA, just a different number of, different sizes of chromosomes. No change in chromosome number.
Could you possibly get a combination of a small arm and a long arm instead of?
Sure. So, yeah, you can do any sort of thing. Well, nature can do any sort of thing. So it could, for example, let's see, different color? Fuse that small arm to that long arm. And then you might have that sort of a fusion. So all of these are reciprocal translocations. Chromosomes break and they switch, different chromosomes reattach to each other in new combinations. This also happens relatively frequently in the evolution of genomes. What causes the DNA to break? What causes chromosomes to break? All sorts of things. Ionizing radiation, ultraviolet light. Errors during mitosis. Microtubules grab the wrong parts of chromosomes. They actually physically pull the chromosomes apart. That causes chromosome breakage. So chromosome breaks happen pretty frequently. Chromosomal rearrangements happen pretty frequently.
In genetics, you had mentioned that telomeres shorten with age. Would those centromeres ever break because of the shortening, or is that just exclusive to telomeres?
Right, so telomeres do shorten over time, but we have enzymes in our cells that keep them long. And, well, in our stem cells, in our germline, we have enzymes that keep our telomeres long so that we don't pass on shorter and shorter telomeres to our kids and grandkids and so forth. But those processes don't occur in centromeres. So centromeres basically, for the most part, stay the same size generation to generation. That's partly true and partly a lie. I'm just trying to simplify. Centromeres are very interesting, just like telomeres are very interesting. They're special parts of our chromosomes that do weird things. So what scientists did to really generate proof beyond chromosome banding that our chromosome 2 is made up of 2 fused primate, non-human primate chromosomes was use fluorescence in situ hybridization, which will be familiar to many of you. But here's the concept. So say you want to know where a gene is physically located on a chromosome. So you take a copy of that gene, the double-stranded DNA that encodes that gene, and you go through this process that's not important, the details aren't important, to replace some of the nucleotides in that gene with nucleotides that have labels on them, special chemical bits. In this case, they're talking about digoxygenin, dig, D-I-G. So it's a particular chemical that's attached to, in this case, uracil, D-U-T-P. So you've incorporated some special chemicals into the copy of the gene you're interested in finding where it's located. And that produces what we've referred to in the past and will continue to refer to, wow, that's a fancy color, as a probe. So it's a labeled piece of gene that we can track, we can visualize where it goes when it attaches by DNA base pairing to a chromosome. So separately, so that's preparing the probe on the left side. So the important thing is you have a gene that you want to know where it's located. You chemically treat it so that you can actually see visually where that gene is. On the other side of the slide here is preparing the chromosomes that we're going to apply that probe to. You take a cell, you break it open, you make sure that the chromosomes in the nucleus adhere to a glass slide. So fixed cells on slides. So this is cytoplasm. The darker blue is the nucleus. And then that probe DNA sequence hybridizes by base pairing to what's shown here is a chromosome stuck to the glass slide. So you use a microscope to actually look at where do you see fluorescence, in this case, the label that's on the probe, the DNA, the gene that you're looking for. So you can see what part of the chromosome contains the gene. Which chromosome is it? You tell that by the size of the chromosome, where the centromere is, the banding pattern. You can see where this gene is located. So now I'd like you to make a hypothesis. This is what the geneticists did. They said, okay, if we want to prove that human chromosome 2 is related to these non-human primate chromosomes, we think they fuse. How would you use fluorescence in situ hybridization to do that? This is a common technique, FISH, fluorescence in situ hybridization, that previous slide, to teach, to learn, to look at how genomes change and evolve. Because it lets us visualize how bits of chromosomes are related to each other. So what sort of an experiment could you do if you want to know what the relationship is between human chromosome 2 and all of the non-human primate chromosomes that we think make up our chromosome 2? So let's say you know that there's a gene on chromosome 2 in humans. What would you do?
You insert the probe exactly on chromosome 2.
So let's say human chromosome 2 has gene A. So you make a FISH probe that contains the human version of gene A. Where are you going to find this gene? Where do you expect to find this gene in chimpanzees? If this relationship is correct, we'd expect to see it in the same area. So if we see fluorescence of this gene A on chromosome 2 in humans, we might predict that we'd see it in exactly the same spot on all the related chromosomes. So what would you have to do regarding the Q arm here? So that suggests, if we saw that, that would suggest that that part of our chromosome 2 is related to those non-human primate chromosomes. Where did the long arm of our chromosome 2 come from? Right, so you'd want to have some gene down here, like gene B, a gene that's known to be on the long arm of human chromosome 2, create a fish probe, and then hybridize that fish probe to the non-human primate chromosomes and ask, where do you see them? If they look like they're all in the same spot, then you've proven the relationship between, at least one gene's relationship between where it's located in our chromosomes and where it's located in our close relatives' chromosomes. Alternatively, if you happen to pick a chromosome or a gene like gene C here that's on our long arm of chromosome 2, and you saw this, where that gene happens to be on chimpanzee chromosomes, whatever those are. Yeah, 7. Then that would suggest a different hypothesis about the origin of the long arm of our chromosome 2. Maybe that would mean that it's chromosome 7 that fused to another chromosome to make our chromosome 2. So a lot of fish studies, fluorescence in situ hybridization studies, use multiple probe sequences to elucidate all of the relationships between all of the chromosomes in different species. We can do it more easily than that. And this is where this experiment really got convincing to biologists and to the general public, assuming the general public knows how fluorescence in situ hybridization works. What would happen if you chose a single fish probe, a single gene that's found right here where our chromosome, we think, fused? So draw on your slide, if you want. If our chromosome, we think the fusion site is right there, inside this fish probe, and the fish probe is labeled red, so we see one big whopping red fluorescent signal corresponding to that fish probe on our chromosome 2, what would this look like when you look at chimpanzees? Right, so half of that fish probe would be located on one primate chromosome, and the other half of it would be located on a different chromosome. If, assuming your probe is really big, talking about a whole, not just a gene, but a huge part of a chromosome. It could be 1 million base pairs. More likely something like 200,000 base pairs, which is about the normal size of a fish probe. It contains multiple genes. So that's exactly what was done in 2000. They happened to find, a lot of work went into finding this probe. So what they did was they took probes from all across chromosome 2. So they pick one that's here, and one that's there, 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 and they take a bunch of human chromosomes and a bunch of non-human primate chromosomes, and they just check, which part of our chromosome 2 do we see this hybridization pattern on the right? where that probe identifies a single spot on our chromosome 2, but it hybridizes to the ends of 2 different chimpanzee chromosomes. So that's how they knew exactly where on our chromosome 2 chimpanzee chromosomes fused. This FISH probe is at a single locus in our chromosome 2 and 2 different spots on chimpanzee chromosomes tells us that those chromosomes fused inside that region. So now we know exactly, essentially down to the nucleotide, where the fusion spot on our chromosome 2 is, where ancestral chromosomes stuck together to make what we currently have as chromosome 2.
Were there any follow-ups where they did any sort of PCR and gel to see the lengths of DNA that matched?
Get down to the nucleotide? Yes. Yeah, so you can go from fluorescence and situ hybridization. If you're really interested in figuring out where exactly the chromosomes fused, you can go even higher resolution using different techniques. So to be a little bit more explicit, if we have human chromosome 2, Homo sapiens HSA, chromosome 2, if we have a FISH probe that hybridizes across part of our human chromosome 2, and if we're saying that chimpanzee chromosomes are identical in sequence to our human chromosome 2, or very similar in sequence, then that same FISH probe would still hybridize to part of the top chromosome, and other copies of that same FISH probe would hybridize, say, here to the top 2 nucleotides on that chromosome, and then there'd be some nucleotides hanging off into space here that didn't hybridize to anything. So you have a perfect match across the human probe and the human chromosome by definition because the probe came from human chromosome. So the top part of this probe hybridizes to the one end of one chromosome, and the bottom part will hybridize to a second chromosome. And that's how you get what you see here. It involves luck, basically. Finding the probe, the particular section of human chromosome 2 that exhibits this trait. And this is done in a lot of species. It's just a useful example to talk about evolution. Questions, concerns? This is something else you can do with fluorescence, which is even more powerful because it doesn't require you first to figure out which specific chunk of chromosome you want to use as a probe. You just take an entire chromosome out of an organism, label the whole thing fluorescently, and then see where it hybridizes to chromosomes from other species. So for example, here are 5 different human chromosomes that are labeled 5 different colors, and they threw all of those chromosomes onto a human chromosome spread. You see that there are 2 chromosomes that are chromosome 3 colored. Right? There are 2 chromosome 1s, which you'd expect because this is the control experiment. You've taken human chromosomes, you fluorescently labeled them, you put them on human chromosome spreads, you see that 1-to-1 relationship. So how many chromosomes from Chlorocebus comprise human chromosome 5? If you take human chromosome 5 as FISH probes and look at where they're located in a primate, it looks like there's 1 pair of chromosomes that are that color here and another pair here. So those must have fused together to make what is now our larger human chromosome 5. So this is looking over greater evolutionary distances than human and chimp, human and old world primate. So human to chimp, 1 chromosome fusion differs between us. Human to more distantly related organisms, lots of chromosome fusions occurred in genome evolution. So don't bother with this fish exercise. I want to leave you with something to think about for next class, which will be Friday. Everybody's, or most of you heard about Zika virus recently in the news. So supposedly there's a relationship between Zika virus and microcephaly, abnormally small brain size. And so what we move to next class, the end of the genomics or the genome evolution chapter, is how we figure out what genes might be responsible for changes like the changes in brain size we've seen in the evolution of humans. So our distant ancestors had really small brains, and in the very recent past there's been a distinct growth in the size of our brains. So how do we figure out what genes are responsible for things like changes in brain size? What I'd like you to do for next class, come prepared to discuss. Here are a number of genes in humans. Pick one of them, Google it, look it up on Wikipedia, look it up on omim.org, and come prepared to talk about what does this gene do, pick one at random, and why you think that that gene might be important in how modern humans have evolved recently. And we'll start next class with that. Thank you.